बंद करो, ये देखो। Dear all friends from Brazil, good morning, and all my friends in India, good evening. At the outset, I, Dr. Shyam Zawar, National Mentor for IVF Labs in India, appointed by Department of Animal Husbandry and Dairying, Ministry of Fisheries, Animal Husbandry and Dairying, Government of India, on behalf of Bovine Embryo Transfer Group of India, welcome you all and our esteemed speaker from Brazil, Dr. Yeda Watanabe. Before we proceed further, I invite your kind attention for following important points. First one, that I request all of you to keep your audio muted during the webinar. While the talk of the guest speaker is in progress, if you have any questions, please use the chat box which is there on your Zoom screen and post your question. We are also doing live stream of this webinar through my Facebook account, so other participants can watch the same. This is just for your information. Now, with this note, let me introduce our guest speaker, Dr. Yeda Watanabe. She is having master's degree in genetics and animal breeding. She is doctorate in genetics and postdoctorate in gene expression of IVF embryos. She is one of the renowned embryologists in the world. She was a coordinator for commercial production of clones. And then she is also a principal thematic researcher in gift genomic breeding. She was forwarding the functional placement and scientific director from 1998 to 2019. You will be happy to know that she is the first one in Brazil to start commercial embryo transfer well back in 1998. So we are proud of her to have her as our guest speaker today. She is involved for coordination, implementation of several laboratories and commercial laboratories around the world, like in Brazil, Colombia, Venezuela, Uruguay, Argentina, España, Australia, Mexico, India, Russia, and Peru, and so many other countries are there. She has published many scientific articles. Now, before I request our guest speaker to deliver her speech, let me take this opportunity to introduce you the members of core committee or central monitoring unit for the IVF labs in India, which is being constituted by Ministry of Animal Husbandry, Government of India. I will request my colleagues, the members, 5 members, whenever I say their name, their video is on, so they can just wave their hands so others will know about them. So to start with, about this central core committee, as I told you, I'm heading this committee, and Dr. Mutharao is one of our members. He is a professor and head at the SV Veterinary University Livestock Research Station, Lamb Farm, Guntur. He is doctorate in animal reproduction with 30 years of research experience in embryo transfer, particularly in buffaloes and indigenous cattle. He is presently heading a IVF lab, which is established at Lamb Farm, Guntur, and from this lab already 15 IVF calves are already produced. Our next member is Dr. Sanjeev Chawal. He is based in United States and he has joined us from USA. He did his doctorate from University of Connecticut, United States, overall having an experience of 35 years, almost out of which 20 years he has spent in ET work in United States, working with a leading company in United States known as the Transbova Genetics. Our next colleague is Dr. Ajay Aswal. I don't know whether he has joined or about to join. He is master's in biotechnology from NDRI Karnal with a 16 years experience with ULDB. And he has an excellent experience into the MOET and IVF. The fourth of our member is Dr. Siddharth Laik. I think he also is probably on the way to join. He is a doctorate from the NDRI Karnal and a Fulbright fellow from Cornell University, working with NDDB for the last 7 years. And the next is Dr. Amol Sahare, who is also doctorate in animal biotechnology with having a 6 years experience into the IVF. And I myself would like to say 2 lines about me. I'm also doctorate in animal reproduction well back of 1990. And mine was the first PhD thesis on superovulation in sheep, goat, and cattle in the country. And I had about 46 years of experience in all over sheep, goat, and cattle. And still I continue with my interest into this technology. Now, let me have all of you, as you know, last week, you have all heard about the guest speaker, Dr. Rodolfo Bonilla, about the important tips to get the maximum number of oocytes from the OPU. Now, today, our guest speaker, Dr. Yeda, will give us important tips as to how to get maximum number of IVF embryos from these oocytes. I mean, to have the best and the best possible blastocyst rate. And also, she will focus on the freezing of embryos, which is a very important in the context of India also. And so in view of this, with this introduction, I request Dr. Yeda to start her presentation, please. Over to you, Dr. Yeda. I just wanted to interrupt for a minute. Dr. Yeda, basically, she is settled in Brazil, but she is from Japan, and somehow her English is little, is understandable, but in case if you have any difficulty, I will help you, no problem. So we welcome this great scientist and a great embryologist known around the world. And I think every one of you know by her product, that is Reproduction, which many of you are using in your laboratory to get a good blastocyst rate. Over to you, Dr. Yeda.
Yeah. Okay. Thank you for inviting me, Dr. Shan, and everybody. And I hope that you can understand. My English is not so good, but I will try to pass all the information to you. And at the end of the presentation, we can talk about all the questions, okay? Now I need to
share your presentation, yeah.
Where is it? This is okay?
Yeah, okay.
And the sound, the audio, it's okay?
Yes, all okay. You put this into PPT mode? Is it in PPT mode?
PP.
Yeah,
PP.
okay.
Yeah.
Good. Presentation. Please go ahead, Yeda. Thank you.
Okay. I will talk with you about the IVF embryo production and how can I improve the quality of these embryos to do the cryopreservation. Then I will talk some steps, all this proceeding to produce the embryo. But to start now, why you can use the commercial, the ovum pickup? Then if you understand to produce the embryo with this technology, it's only to increase the embryo production. Because if you compare all the another biotechnology like insemination artificial, you can produce 1 pregnancy per year, okay? And if you use the MOET, it's a multiple ovulation embryo transfer, the superovulation, you can produce about the 20 until 30 pregnancies per year, okay? Because you can do maybe the 5 or 6 flushing in a year, and if you have 6 pregnancies per flushing, then you can produce this. And I will show to you how can how the IVF is possible to produce more embryos or pregnancy. Okay, so then you can produce the 2 or 3 pregnancy for open section, the OPU, the aspiration. And in 1 year, you can produce until 70 pregnancy. One cow, then you can produce all this. Another that you can use is sorted semen, because if you work with a beef cattle or dairy to produce milk, then you can use the semen sorted to male or female, and you can use 1 straw to do the mating, the 8 or 10 donors. This technology, it's not necessary to use the hormones to superovulation or use it to stimulation the donor to do the ovum pickup. Then you can do all the proceed in the ovum pickup, all the oestral cycle. Then it's not necessary to stimulate it. And of course, if you work with genomics, if you identify donors, heifer, prepuber, then you can produce the embryo too. I think that tomorrow, Professor Pietro will talk about this different age of the cow that you can produce the embryos. Okay, and the cryopreservation of the embryos. This is the history of the IVF in bovine in the world. The first calf born, the first calf, the IVF born in 1981 in United States by Brackley. In Brazil, during my master in Jabuticabal with my professor, this is my professor Enoch in UNESCO in Jabuticabal and me, and this is the 3 calves that born in Brazil in 1992. This is the first calf that born in Brazil with the IVF. Then when I finished my doctor in 1998, we started a commercial IVF lab here in city de Cravinhos, near to Ribeirão Preto, São Paulo, and 24 years ago. After 5 years in 2003, WTA started to development all the kind of the product to IVF and the equipment too in vet and in human. Start 4 years ago to produce this equipment for the IVF in human too. Now in Brazil, you have almost 50 commercial IVF labs, because now I will show what is necessary to produce the embryo. Now you have a small lab, and it's possible when you compare all the equipment that you have in the past and today, what kind of the equipment that is necessary to produce embryos. Now, all the steps that I will talk about the system, the production, the embryo, I will talk about these steps, the production, the embryos, some factor that can interfere of this production, like donor, bulls, and some project that you development here in Brazil or another country too, for beef and milk, and all the equipment and to logistic and improve the equipment to produce the best embryos and media too. On equipment and the medium you develop this. And the cryopreservation of the embryo, some result compare vitrification and DT freezing. This is the steps. This is the steps. The OPU. Then in the first day, the last one that you see, like Monday, you need to do the ovum pickup. Then you do the ovum pickup in the cow, select the oocyte, and this oocyte you put in this vial with the IVM medium and put in this transportation of the oocyte, the lab mix oocyte. This lab mix has a gas inside of this equipment. Then you can arrive in the lab the next day to do the IVF. Then you need to prepare the semen and put together the oocyte maturated, and you have all kind of this medium to do this, prepare the spermatozoa and to do the insemination. And another day, in Monday, start with the ovum pickup, Tuesday, IVF, Wednesday, start the first day of the culture, the embryo. Then stay there in this day 1 until day 7 to produce the blastocyst. Then you put inside this benchtop incubator, the 1 cell zygote until blastocyst. In this day 7, you can do the transfer of these embryos, fresh transfer, or cryopreservation by technique, the vitrification or DT freezing. For this step, the ovum pickup, this is in the farm, the vet, here's the cow contained here in this trunk, I don't know in English. They put this, the cow here, the ultrasound, then you need to take the ovary, and this is the probe that which connect the ultrasound and the tube, in this tube that you receive the oocyte. Then do the aspiration. Then you have 2 kinds of the handle. You have this size, the handle. This is the handle, it's big here, and then another is the small. This it's possible to work with the heifers, and this, some heifer you can use it, but sometimes it's not enter in the vagina in the cow. Then you need to use the small, like this. Here you have the vacuum pump with the arm and tube, with the medium to do the ovum pickup. Then you put here in the vagina, you can see here, and cross the fundo of this, and this is the ovary, and to do the ovum pickup. And here in the slaughterhouse ovary, when you receive the ovary in the lab, and do this aspiration, and you have all the oocyte and put it to maturation. You can saw this is the ovary that you saw in the ultrasound, and you can see here different follicle. Tomorrow, I think Pietro will talk about all this size of the follicle, the competence of this follicle to do the ovum pickup. And in the farm, you need to prepare the room to do the selected oocyte. Okay, then you need to do the stereo microscope, the lab mix, and the Warman table plate here, and the pipettes and everything to do the select this oocyte. And you can see here all this. This is the equipment and material used for selecting the oocyte. And when you receive the tube, aspirate the donor here, this is the tube, the 50 ml with the blood and the PBS and media, then you can put in the filter here. Here, you put in this size of the filter. This is the filter. You have two kinds of the filter. This is the filter to IVF and have another for embryo transfer. And this is the big filter. Now you use this small filter, the mini filter that you can use here. You put all the filter, the aspiration here, washing this and put in the plate. And go to the stereo microscope to select the oocyte, put inside this vial, the micro vial, with a 500 ml the IVM media and with oil, and cut here and put inside this incubator. Then you can arrive in the lab the next day after 24 hours to do the ovum, to do the fertilization. Okay? Então, this is the step, the in vitro maturation for 1 day, 24 hours. This is the communal cells and expand these communal cells. The IVF, you need to prepare the spermatozoa. You put the semen here. And this is the Percol gradient, centrifugate this, and the viable spermatozoa get here, and you take this and to put in the drops with the oocyte to do the insemination. And after 3 days, you have the cleavage embryos and the blastocyst here. This is the blast that you use to transfer or cryopreservation. Now, I will talk about what kind of the interference that you have in this production of the embryos. Some factor, biological factor, like variation among the donors or the genetic groups, Taurino and Zebuino, Indicus or Taurus, variation among the sires, and the interaction between sires and donors. This is some results when you start in 1998, and you have the pregnancy rate about the 80% of the blastocyst of the pregnancy, and the 32% rate of the blastocyst, and now you have the result more stable and the pregnancy too. This is the variation. The average, the production, the embryo, I talk about the 35% of the embryo in almost 40% of the pregnancy. And you can see here different cows, 18 cows, and the quantity of the blastocyst, the embryo for OPO and donor. This is the average. Some donors produce 13 embryos for aspiration, and another 1 or 0.1, 0.2 embryos for aspiration. Then you have some donors produce more or less embryo for aspiration. And pregnancy too, you have the variation between donors, between cows. This is a cows, Nelore cows, that you produce in only one, in the first aspiration, you produce this quantity of the oocyte, 437 oocyte, viable oocyte. Then you use mating with six different bulls, and you produce 41% of the blastocyst, 181. In this case, in this year, I don't remember the year, but I think it's 15 or more, 18 years ago, you don't have the technique of the cryopreservation, vitrification, then you don't have the recipient enough and you transfer only this quantity of the embryos. Then you produce the pregnancy, 64 for pregnancy. Okay, this is the cow that produces a lot of oocyte and embryos and pregnancy. And this donor, the next time that you do the aspiration again, the secondary aspiration, you aspirate only 1 ovary because this donor was pregnant, and you produce more 39 pregnancies. Then you can see some donor produce a lot of embryos and another no. This is a study, retrospective study that you work, Vitrogen, the equipe, and the Pietro equipe, the Pietro in São Paulo, to study all the data, the Vitrogen for the 2001, 16 years. You take all the information, the Vitrogen, and compare the production, the embryo and the oocyte in Nelore, Gyr, and for Nelore, Gyr, Senepal, and Holstein. 2 Indicus and 2 Taurus for milk or I forgot, milk and meat. And the total of the donors that you aspirated, this is the quantity of, from beef and there. Nelore, Senepal, Gyr, and Holstein. The quantity of the donors and the quantity of the oocyte each breed. About 8,000 proceed in the OPU with different veterinary and the quantity of the oocyte here. Then you can see for Nelore here, you can see here, if you divide in four steps or quartile, then you can have some quartile, the less quantity of the oocyte and the more quantity of the oocyte. And the average for Nelore is 27 oocyte per PU. Senepal, the average 22, almost 22. Gyr, 24, and Holstein, 19. Then you divide all these in for the quartile lower, intermediate, and the superior quartile. Then you'd like to know if some donors that produce less oocyte, the quality of this oocyte to produce the embryo is the same or not. Then you can see here the production, the embryos, the rate to produce the blastocyst is similar. Then the quantity, it's not correlated with the quality of this oocyte to produce the embryo. Of course, that produce more embryos here because you have more oocyte. Then the quartile, the superior quartile produce more oocyte, and the quality is the same for the Nelore breed. This is the Senepol, another breed, Bostaurus beef. Gear for milk, you have some donor to produce the average oocyte 8 until almost 50 oocytes. And Holstein too. You have here some cows, 169 to produce the average almost 40 oocytes per ovum pickup. And this is the quantity of the embryos. Then you can do, and Pietro and Fabio Monteiro have a work article that this is have a great repeatability. Then you can, these cows that you select to produce more oocyte, the daughter can produce more oocyte too. This is the quantity of the bulls. You can see here, you work with 5, oh, sorry, wait a minute. We work with a 48 bulls, and some bulls produce less blastocyst and another more blastocyst, almost 60% of the blastocyst and sometimes 15% of the blastocyst. Then it's very, very important that you have a bull in your lab if you work with research and everything, and you need to take a bull to produce and the standard of the lab. Here is the different bull, 90 bull. This is the quantity of the cows, the repeatability that I use in the IVF commercial. The quantity of the embryos, here the blastocyst and the rate. This is the average 35% of the blastocyst. Some bull produce less blastocyst and another produce more blastocyst. And here the pregnancy. Then you have the pregnancy more high here, and sometimes like this bull, the F, the bull letter F, produce almost 28% of the pregnancy, less than the average. And you can say, oh, this bull, it's not good to do the pregnancy. But if you compare, this is the bull, and if you use this bull with different cows, this is the quantity of the embryos transfer. Then you can saw this, the donor 2 produces 75% of the pregnancy, and the another 4, donor 4, almost 43% of the pregnancy. Okay, the problem was in the donor 6 because I transfer a lot of embryos and the pregnancy rate is lower, 28%. Okay, but this donor 6, if when I use it with the another bull, this bull H, then you can produce more embryo. Then you need to select the best mating, this cow with this bull to produce more pregnancy. Then you need to work with this information. Here, this is a project that you start in the years 2001, in 2 years. And Senepal, this is the breed Senepal, Taurus, to produce beef. And the first year that we work 3 months, 90 days, and you produce this. 30% of the blastocyst. This is the quantity of the oocyte in the phase 1, in the 1 year, and the pregnancy rate was 26% of the pregnancy. Then the next year that you have all the mating, the donor with use another bull, I choose the best mating, the best interaction cow and bull, and you can see here that improve a lot the pregnancy rate. Then you have some interaction that result in more pregnancy. Then you need to work with this kind of the information too. Then you can see here, when you start the IVF in Brazil, during 16 years, you can improve the pregnancy per ovum pickup, the 1 until 2.4. This is all the breeds that you work here, in these numbers. Because improve a lot of the protocol to do the embryo transfer, the synchronize the recipient, the quality of this, the quality of the medium, the system of the production, the embryo. In these years, you have increased a lot the quality of the embryos in vitro production. You have some project, the beef and milk. This is the project, the Senepal, that you start in this year, 2000, 2001, in north of the Brazil. And because you don't have technique don't have the cryopreservation embryo yet, only transfer the embryo fresh. Then you need to do the IVF lab near to donor and the recipient. Then you produce, this is the first phase of this project, then Senepal in the farm. In 3 months, you change the breed in the farm to produce all this kind of the breed, the Senepal breed. Then the WTA developed the equipment to, this is the equipment, the lab mix that now you can, it's possible to have the donor distance of the lab, because you do the ovum pickup in Monday in the farm and put the oocyte here, and during the transport until the lab the next day to do the IVF, you have all the system, this incubator, portable incubator to bring the oocyte to the lab. And you can transportation these embryos, cleavage embryos, in day 4 or 5 or 6 until day 7 to start the embryo transfer in the farm again. Então, this is the logistic, and this is the another industry of the production, the embryo, Nelore embryo in this case. And this logistic, you produce the embryos here in Uberaba, okay? Don't have yet the cryopreservation of the embryo. Then you need you did the ovum pickup in Monday, Tuesday, Wednesday. 3 days the ovum pickup sequence. The another week, I put the embryos in Monday, I put the embryos in incubator, the lab mix incubator, the embryos in day 4, 5, and 6, and go to another, the north of the Brazil, 2,000 kilometers, the distance, and transfer the embryo. They say until day 7, start, you arrive in the farm Monday, and Tuesday, transfer this embryo, Wednesday, transfer the embryos that stay in the incubator for 2 days and another for 3 days. Then you can saw the result of the pregnancy was very good in the both, in the all this time period that the embryos stay in the lab mix. Then yet it's not possible to cryopreservation the embryos. Then you can do this, all this logistic to transfer the embryo fresh, the fresh embryos. This is another project that you would like to do in California with hosting, with the vitrified embryos. Then you take all the oocyte, the slaughterhouse ovary during winter, yeah, because the stress thermic of the ovary, the oocyte, it's not so good. Then the idea is produce the embryos during the winter, vitrify these embryos, and this is our lab in California with this incubator, 2 incubator. You can saw here, put the oocyte here, and do the vitrification. And when start the season with very hot, because this is not so good in the summer, it's not so good for the quality of the oocyte, and you use the embryo transfer. This technique is, the name is embryotherapy. Okay, this is the results. When you use some breed Holstein and Angus, you produce this quantity of the oocyte, the embryos to vitrify, and Angus too. And this is the variation, depends of the bull that you use with this ovary slaughterhouse. You have a lot of variation here. Like this bull produce 10% and the another 40%. This is the work, the Barrucelli, the Pietro Barrucelli, that in Holstein. And if you did, this is the insemination and the embryo transfer. Okay, if you do the embryo transfer, the insemination during the summer, the result of the pregnancy is lower and different quantitative when you do the embryo transfer. Then the idea is during the summer, I use the embryo, vitrify embryos to transfer the embryos, and you don't have the difference during all the season, the summer, autumn, spring, and everything, because the oocyte the stress is terrible for the oocyte, not for embryos. This is the production in North America. Like in North America, you have, this is the quantity of the IVF embryos, but this is 80 years ago. And you have 25% of the blastocyst, and in South America, because you have a lot of Nelore produce more oocyte and more embryo, then the average is high compared with Boston's. And now I will talk about the logistics. This in the past used this kind of the warm water to keep it warm the vial and put in this thermical. Now you put in this warm plate, and you don't have water, because the water in this kind of the system, you can contamination. You can have a contamination. This is the select, and the oocyte put here in the holding medium. Now you can put in the IVM medium and put the incubator, portable incubator. Then you can arrive in the lab another day to do the insemination. And here, when you arrive in the lab at night, then you need to put inside the drops the maturation and put the incubator. Now it's more easy. You develop all this kind of the equipment to facilitate and improve the results too. And like this, the big incubator, now you have a more stable, this incubator, the benchtop incubator. And in the past, you used this to transfer the box, this kind of the box, to transportation of the embryos inside the straws. Now you have all this kind of the transportation, the embryos. So this is the step that you use different kind of the equipment that development to facilitate the increase the quality of the embryos. Sorry. When you compare the big incubator and the small incubator, then you can work in the less space in the lab to produce this embryo. Now I will talk about because when you use this kind of the equipment, you work with low tension of the oxygen. Okay, you can use the premixture of the gas already, and the quantity of the oxygen, you can choose what the quantity that you want. You put all the oocyte here, and this is when you start to development this kind of the equipment. You compare the product with your slaughterhouse, the product the embryo 40%, compare the another group in the big incubator. And the result is similar, the production the embryo. Like here, the incubator, the big incubator, or the EV incubator, or benchtop or conventional. The production here, it's more. And the problem is when you have a big incubator and open the door here, during 15 minutes, you open the door, wait 15 minutes to take the plate and close the door. You have 1 hour, almost 1 hour to stabilize all the humidity, temperature, and the gas. And in this incubator, it's very small, and the equilibration, all this variable, it's very quickly. Then you can put here inside this plate, different kind of the plate and the tubes too. When you start some lab in Brazil, using this kind of the equipment, and this is the different labs in Brazil that have this kind, and this is the production, the embryo transfer, and the pregnancy. Then you can saw, this is the result, it's very good, the pregnancy in 60 days. And in this case, in this table, you use the 5% of the oxygen during the IVM and the IVC. Then I'd like to compare to use all the steps 5% of the oxygen, because physiological, the concentration of the oxygen is lower than 20%. This is the quantity of the oxygen that you have in the air outside. Okay, then the production, the embryos is lower, but the quality of these embryos is good. Like here, you can see the pregnancy rate. These years in the ITS group of Peru work with this and have results good, working with lower concentration of the oxygen during the IVF. This is the lab when you use big in the past, when you use the big incubation. This is it's necessary more space. This is you put the tank here, the IVF lab here the medium to prepare the medium, wash and reception. And you can tell this is inside of this lab. It's very big. It's not necessary today this, because you have 2 incubator, laminar flower. This is kind of the centrifugal. No, you don't use more this kind of the centrifugal to prepare the semen. Now you use the mini percol, and this is the small. This is the Mexico, our client Mexico. Then you can saw this is the lab, the IVF here. This is with refrigerator, something, and this is the reception. This is the small lab, but you have the big incubator here. But this, if you compare, this is the centrifugal that you use now, not more this kind of the centrifugal. And the proposal is to use the small lab, the compact lab, today to produce almost 2,000 embryos per month. If you have 2 incubator and this laminar flow, stereo microscope, and centrifugal, it's enough to produce these embryos. 2,000 embryos per month with this kind of the equipment. Only this. 2 incubator, the centrifugal, microscope, stereo microscope, flower, laminar, and refrigerator. And this is the autoclave to do some sterilization, the tips or something. Now I'll talk about the cryopreservation, the IVF, and compare some results. And to do, how can, to do the cryopreservation of these embryos, you need to think, I need to produce the embryos the best quality. And how can I do this? How can I improve the quality of these embryos to produce embryos enough to freeze or vitrification or to do the DT freezing, okay? Then you need to produce the embryos more similar to the physiological, chemical, and the physical principles, okay? And you need to do this, development some media, equipment, and everything, okay? Then you start to work with this almost 4 years with a project here in Brazil. And you need to take a pH ideal, it's good pH, it's about this, 7.3 or 4, lower concentration of the oxygen, and with the micro vibration, okay? Because inside the oviduct, you have a ciliary, that you have a battement ciliary, that you have some movement. Then you have some result with this in human, and I will show. And in our results too. Like a pH change when you have, of course, outside of the incubator. But if you use oil in your system to produce the embryos or drops or 5-well dish with oil, then you can maintain more stable the pH when you don't have pH, because when you take the plate outside of the incubator, the pH of them may increase. Then you need to take care with this. If you open many times the incubator, in this case, if you open every day and several with more frequency during the day, then the pH it's irregular, okay? Here, this is a group in the human that you work a concentration of the oxygen during. This is all the gametas, oocytes, spermatozoa, and embryo stay in the incubator with 20% of the oxygen. Here, the gamete is 20, then put 5% of the oxygen, or only in 5% of the oxygen. This is the quantity of the cycles, the age of the human, okay? And here, the number of the embryo transferred, that you transferred for cycle. Then you can compare the quantity of the embryos, the number is the same in these 3 groups. Okay, then it don't have difference in the production of the embryo. But if you have the pregnancy rate here is high in the group that you maintain all the gametas and oocyte with 5% of oxygen. Then you need to use lower oxygen because the stress, oxidative stress. And this is our results with our clients that have a big incubator, then work only the 20% of the oxygen, high percent of the oxygen, you can produce more embryo, and the pregnancy is here too. And when you use all the client 2 or 3, all the step lower oxygen, you can produce less embryos, but the quality of the embryo is good. And in this case, you use high oxygen during the IVF, and you have this. When you compare here with 5% of the oxygen, you produce less embryo, but the pregnancy rate is high when you compare the another group that you use 20% of the oxygen. It's like the another result in human. Sometimes the production, the embryos, the same, but the quality of these embryos is high when you use the oxygen more similar to physiological. And another characteristic, it's a mechanical microvibration, because the culture, the in vitro culture, you can compare the static versus dynamic, because inside of the oviduct is dynamical, and you have abattimento ciliar, the frequency of the ciliar beating, it's about the 5 to 20 hertz. Okay, and this is very, and increase this beating in the region, the ampulla and the isthmus. Okay. Then this is a work in human that compare static, and this is the group with this microvibration with co-culture, okay? With some co-cultures with its cells, the oviduct cells. And here you can see the pregnancy rate increase when you have compare the static culture of these embryos. You can see here the quality of the embryos frozen. In this group, the dynamic group is more high than static group. Like this, the cumulative pregnancy, it's high compared to the static group. All this is in human. And this is our project. This is the incubator, the normal incubator, and this is the incubator, the modular incubator, EV, that you have some vibration and pressure inside the, it's a pressure and vibration with this kind of the equipment, the incubator. The idea is you put here only to see the distribution of the molecular or the nutrients. Then if you use, this is the time 0, and after 1 hour and a half, you can saw the distribution of this color, right? This is the color, only to see the distribution of the nutrients. And this is the graphic of the microvibration. You do the, after 30 minutes, you have a peak of the vibration during 5 seconds. And you do this. And you can see the production of the embryo with, this is the maturation, using the vibration maturation, and or in culture IVC, in the culture embryo. And you can see this is lower when you use the maturation, but when you use the vibration during the culture of the embryo, that happen this in oviduct during the transport of the oocyte, the ampulla until the uterus to pregnancy. Then you can see the result is bad, is better when you use the in the step of the culture. This is different pressure that you use inside the incubator, and you can see the results with this, the blastocyst in day 7. Then this is the control, this is the 20, and here I'd like to use it, because if you use the pressure inside the incubator, you can, the flow of the gas inside the medium is better. Here the difference is not, the difference is not significant, but I do this is not significant, but here you have some significant, and use this kind of more high pressure. Then you do the conjugated, the pressure and the vibration. This is the pressure and vibration. This is the control. You can see this, you can produce more embryo when you do the vibration and pressure. And here with 2030 seconds, 30 minutes. And you can saw, this is the different blastocysts produced in these groups. And you did some total of the cells in the blastocyst, and you have some more blastocysts in this group with pressure and vibration. And when I do the tunnel to do the apoptotic cells, you can see the more apoptotic cells in the control group, then you use this kind. And this is the pregnancy rate, okay? This is the embryo, the vitrification embryo. You use more, here in Brazil, more vitrification to Nelore, okay? You use this because this technique yet it's more stable, and you have the result, the 40% of the pregnancy with the vitrification embryo. And this is the group that you development, the control, and with pressure and vibration. Then you can saw the pregnancy is the control 32, and this is 37, increasing the pregnancy. But it's not significant because you start this research, okay? And this is the different in heifers, the 12 monthly, 25, and in the cows. And this is the pregnancy rate, the project with Nelore, you produce, this is the 4th time that you use this project, because you transfer almost 500 embryo transfer vitrified Nelore, and you have 40% of the pregnancy using this vitrified. This is another project in Russia that you produce in 1 month and a half, almost 3,000 embryos vitrify all these embryos. And you can saw some different bulls, and some bulls produce more embryos than another. And after transfer, these embryos, you have 36% of the pregnancy with the Angus here. And here is the results central of, they have a lot of recipients, the Mineiro embryo. I think that Dr. Xian know the Eduardo, this vet that have a lot of farm to receivers, and they receive the embryos, the more than 20 IVF labs in Brazil. Then you have some results, they compare the MOET, the pregnancy rate and the abortion, and the IVF DT, it's almost 10% less than vitrified, okay? And this is the average here in Brazil. But sometimes you have some results more high in DT, because it start this and start a lot of work. I have a client in the United States that transfer almost 1,000 embryo and from the T, and have 50% of pregnancy with towels. And you can tell during all these 2 decades, you can improve a lot all the steps, the embryos, number of the oocyte per rep, have the number of the oocyte, it's not correlated with the quality of the oocytes, okay? And if you have a lot of oocyte in the oval pickup or less oocyte, the probability to produce the embryo is the same, the blastocyst. And another factor, of course, is different, like a breed, site, donor, interaction, season, and another variables, of course, that nutrition and everything. And you need, of course, to improve all the systems of culture of the embryo with lower oxygen, micro vibration, a lot of things that will improve the equipment and the media to increase the cryotolerance of the IVF embryos. And of course, I'd like to Vitrogen, of course, acknowledge the Vitrogen, WTA, the university with Flávio Meirelles in Pirassununga and São Paulo, Pietro, all the foundation that have some money to do the research here in Brazil, and then of course the farmers and the veterinary. And thanks, this is my address and phone if you want to talk. And you have in now in Vitrogen, you work to train all the person to train you to do the IVF and different medium to produce the embryo, to do the cryopreservation, vitrification, and everything, and every step. The tea and free and the vitrification. Okay, it's enough.
Thank you, Dr. Yeda, for such a wonderful and very illustrious talk. You know, you're like an encyclopedia of embryology. You know, you can there are so many questions coming in for you, but we will give you a break for some time. And meanwhile, I request my panelists, who are my colleagues into this core committee made by the government of India. So I request, first of all, Dr. Sanjeev Chaubal. You know, Dr. Yeda, Sanjeev is based in United States, and he's basically from India, but he's working with Transova Genetics for the last 20 years. And he is an embryologist. So I request Dr. Sanjeev to take about 5 minutes, each of my core committee members, and they can put their views and they can supplement it, but kindly keep yourself for 5 minutes. And after we finish with 5 of you, the comments, then we move to the question and answers, because there are a lot of questions, Yeda, for you to answer it. Meanwhile, you can have a cup of coffee. So we start with Dr. Sanjeev.
Excellent presentation, Dr. Yeda. And the volume of work is just unbelievable. I have only 5 minutes, so I will just comment on 3 points. One is about the incubator size. You have obviously showed that you are using, or you are preferring, the smaller benchtop incubators compared to the box incubators. So a lot of people have question about which one is better. At Transova, we have 7 IVF labs at Transova, and we handle a huge amount of volume. Many of our labs handle 100 to 150 oocytes, 100 to 150 OPUs per day. So for us, using small benchtop incubators is just out of question. And that's the reason we have stuck to the old box type incubators, and they are still working good for us. Secondly, about the lab structures. I was very happy to see finally somebody talking about simple labs, because every time I have visited India and I have seen the labs, they are so huge and so expensively built. In contrast, our labs at Transova in USA and some of the labs that I have seen in Brazil, they are so simple and so modest. I mean, there is just no point wasting a lot of resources and funds on building expensive labs and then not doing enough work to justify them. So I was very model of simple labs. And the third point I was about vitrification of embryos. At Transova, again, we had done a lot of work on vitrification several years ago, and we got very good results. The problem was thawing of vitrification is a little tricky. So long as our own technicians are doing the thawing of vitrified embryos, it is fine. But if you send the embryos, the vitrified embryos, to the farmers and ask them to do the thawing as well, then there is a so to the and that is giving us about 45% pregnancy rate with the frozen embryos. But once again, thanks a lot for an excellent presentation, and it would be an honor to work with you. Thank you.
Dr. Sanjeev, thank you very much, Dr. Sanjeev. Dr. Yeda, as I told you, he's based in USA and he's an embryologist. And then we request another member of our committee who is also an embryologist, Dr. Amol Sahare. Dr. Amol, would you like to make some comments?
Yes, sir. Good morning, Yeda ma'am. As you know that we met in 2019. So I learned a lot from you basically in field level and lab level. So you have presented well in illustrated manner every minute points, that is pH, low oxygen tension, and temperature, especially that micro vibration. You research a lot on that, and the combinations that pressure and vibrations especially affect on embryo production. You study a lot with tunnel assay also. You showed that increase the apoptosis in control and decrease the opposite apoptosis in vibration, your experiment, and increase also pregnancy rate. I like that research. And one my experience is that in vitrification, we culture the embryo that about 2 hours or 6 hours, and we found pregnancy rate around 18 to 20% only. But as per Rudolfo, the previous here the webinar, he said about the just devitrify and transfer the embryo, you get better results. So what is the future of vitrification? Will be future, or DT frozen embryo will future? It will be in a question in chat, you find. So it's nice to hear you, ma'am. After a long time here, thank you very much.
Thank you, Dr. Amol. Yeda, I hope you recollect Dr. Amol, who visited the JK Trust lab. He was there at that lab. Now we are another third embryologist in our group, the core committee group. He is Dr. Siddharth Laik. He is working for a very important organization in the country. You must have heard about it, National Dairy Development Board. So Dr. Siddharth, over to you.
Thank you, sir. We have started our laboratory mainly for training purpose and doing some research and development work. First of all, thank you for a very nice and informative presentation. It's an eye-opener for many of the parts. I'll just add one more point and one declaration. First point is that which we learned from Dr. Chaubal actually is don't rely on the display because display are like display for the incubators, display for the water baths, display for the dry bath, or display for the slide warmers, because they are very deceptive. So always put one more check. Like if your display is saying that gas concentration is 5%, add a gas analyzer. Or if your incubator is saying 38.5, so add a thermometer to it so that you know that you are corroborating that result. So we faced many issues which we corrected with Dr. Chaubal's suggestions. So that's one point I would add on that. And I have a nice declaration to make. That's why I get a little bit delayed because we went to a buffalo farm for pregnancy diagnosis. So our first buffalo endeavor had got success. We successfully established pregnancy in buffalo. I shared in our group previously that we are doing OPU. On 13th, we transferred in December, and now we declare that we have got 2 pregnancies. Thank you. Thank you, sir.
Thank you. Thank you, sir. And hearty congratulations for getting the IVF buffalo pregnancies. Now, I request Dr. Ajay Aswal, who is another member with a rich experience with us, and mainly he has been working with the MOET and also with IVF. Over to you, Dr. Ajay.
Good morning, madam. I'm Dr. Aswal, Ajay Aswal, and I'm working in the state animal husbandry department. We have started a IVF work for last 3 years. Earlier, we were doing, as Dr. Zawar is saying, that we were doing MOET program. And for last 3 years, we have started this IVF program. The only submission I want to say is that in India, we are facing a lot of problem regarding IVF for frozen embryo pregnancies. Fresh embryos, we are getting around 36-37% pregnancy. So that is not a problem. So what would be the future of vitrification direct transfer, vitrification plus direct transfer? So I think that will be a technology which will be very much useful for us in India. So this the only thing I want to say. Thank you, madam.
Thank you, Dr. Ajay. Now our 5th member is Dr. Muttara, again having a very rich experience in MOET and also heading a IVF lab. Over to Dr. Muttara.
Good morning, madam. Good morning, everybody. In fact, thank you very much for a wonderful talk on IVF industry in Brazil.
Professor.
In fact, it's great to know that the Indian breeds, the Ongol and Gir, they are doing excellent for bovine IVF industry in Brazil. In fact, that recollects us of the contribution of the Ongol breed, especially in the state where I am working. In fact, this breed has been doing, this animal we have been working very well for the last 15 years, especially on MOET. We have produced a sizable number of pregnancies caused with this breed. Really great breeds, really God's gift that Nellore breed is. So I am really happy that this Indian breed, especially our breed, is doing excellent in the IVF industry in Brazil. And madam, your talk is really rich. In fact, it's a great learning experience for all of us. And it's a big inspiration for us to move forward regarding IVF in Brazil. In fact, when we are listening to you, this is first time we came to know that WTA means this is a part of your name. So WTA is now a buzzword in IVF industry, even in Brazil. Thank you, madam. Thank you for your all contribution. And we hope we will take this technology forward with this rich expertise and experience and from you. Thank you very much.
Thank you, Dr. Muttara, and for the knowledge of everybody, I will just want to tell you that Yeda, she is a sister of the adjoining is the WTA, so they do test every equipment in Yeda's lab. It's a wonderful lab. I have personally seen that. With that note and with the observations made by our core committee members, now Yeda, we move to the question and answers, and you have a huge list of question and answers. I will read the question very slowly, and then you can give a reply to that. So we start with the first question, that is from Dr. Muttara. What is share of IVF embryos of total produced in Brazil? What is the share total number of embryos in Dr. Yeda?
Yes.
You got my question?
Yes, what is the share of the IVF embryos out? I don't know. Well, the production, the embryos here now in Brazil, I think the last year, you have almost 5,000, 500,000 embryos, yeah. But here to do the exportation in the embryos, it's very difficult to do this. Yes, to exportation. It's very different like in the United States, yeah, because and Canada, and you do this. And here in Brazil, you work, I think the milk and the beef, you start a lot, 10 years start a lot to increase the milk, the production of these embryos like Giroland to produce here inside this, and sometimes go to the another country, the Latin America. Okay. And
there is one, sorry. Next question is from Dr. Siddharth. He is asking if a bull is good in AI, may not be good in IVF. Is it correct?
yes. Sometimes in my doctor, I'd like to work to select the oocyte, because you have some work in the INRA in France that have if you have a bull to produce the embryos, a good producer in the lab have a good production the insemination too. This is select the bull with the IVF. I try to do this, but sometimes it's not possible. It's not possible because sometimes you have some bull that is good in the insemination artificial, but in the IVF it's not so good. And I don't know what's happened with this. If you have some if you want to do some observation about this. Don't know, but sometimes it's different. Some bull is good in the IVF, but it's not for insemination.
Thank you. There is one question from Dr. Ramachandra Reddy. He is asking, Gir cows, hormonal stimulation, how much oocyte you get average from Gir cows?
And in Gir, I have, you produce this, it's almost 20% of the oocyte. This is in the graphic that you will compare Nelore, Gir, Senepol, and Holstein. I think 23 or 24 the oocyte. But you have donors that produce less and another that produce more. Yeah, but the average is about 15, 20 oocyte. And Nelore more, of course, 40.
Yeda, a question from me. What is the maximum, highest number of oocyte from one cow and highest number of pregnancies from one single OPU?
That I show in the slide, yeah? No, I don't understand. What?
In Brazil, what is the maximum number of oocyte
The maximum.
one OPU and maximum pregnancies? I remember you telling that figure. So please tell the participants.
I think you have this. It's about almost 500 oocytes, and they produce 200 embryos, And if you have 40% of the pregnancy, you have another bull. This is the Nelore that I show, but I think that some people here have with Senepol a lot of Elsot, but I don't remember. This is in our lab in Campo Grande. We produce this quantity of the oocytes. Okay, but I think that you have a lot of it now.
Thank you.
But I don't remember more.
Dr. Amol asking you, can we freeze? Will they go after thawing? What is the survival rate? Can we freeze the hatch?
Hatch the blastocyst, yes.
Blastocyst.
Yeah. The problem to freezing or do the vitrification, everything after hatch the blastocyst, it's not so good because the zona pellucida protect of the maintain the cells, the blastoma together, and maintain the structure, and it's more easy to do the cryopreservation. Now, but if you, I do this because because some embryos develop more quickly than another. Then sometimes in day 7, when you arrive in the lab and saw the drop and saw the embryos, and some embryos start the hatching. Now, I do the vitrification or the freezing these embryos in day 6 afternoon, then day 7, and sometimes day 7 or day 8 to, I do this kind of the cryopreservation in 3 times, day 6, 7, and sometimes in 8. And of course, I think the best results is the quality of these embryos inside the zona pellucida and expand the blastocyst or blastocyst or expand blastocyst or expanded blastocyst is the best.
Yeda,
okay, but for fresh you can use, of course, the fresh embryo, transfer this embryo, the result, the pregnancy is good. Do you have some? I remember that you did in the past some blastocysts because it's hatched, and when you cryopreservation, the pregnancy, I don't know what the pregnancy is. I need you to check all these observations. Do you have something, Dr. Ramon?
Dr. Amol, you want to comment? She's asking you.
No, ma'am. I didn't frozen the hatched one. I don't have any data on the pregnancy, so that's why I asked you. You have any data? You have frozen hatched embryo and you got pregnancy.
I don't have this data with this stage of these embryos.
Because in cloned embryo, there are generally we made handmade cloning during my PhD. Yes. Yeah, there is zona free, and we vitrified that embryo, and we got pregnancy. That's why same embryo we can do that.
Yes, of course. In cloning, because they don't have the zona pellucida, and right. I think that some research in the past put this embryo inside the empty zona pellucida to do the freezing or something, but I think it's not, because when you put in the media for the freezing, then you can shrink and can get out of the zona pellucida again. But But I think that the result can have a pregnancy, of course. If you have good embryos, you have the pregnancy, but you don't have the protect of these embryos, because the advantage to do of course, the embryo transfer. I think the embryo transfer DT is the future. You need to work with this, yes, and because you can do the exportation of this embryo because you protect the embryo with the zona pellucida, and you can export it's not necessary the technique like this, all the comments of the veterinary here, because if you have a technique to go to the farm to do the towing, like Transova work a lot, of course, DT. In United States, to work more DT than freezing. Nobody work with the freezing, the vitrification embryo, only the T freezing, and the result is good. And I think with this, you need to have a step of the IVC, the culture of this embryo in lower oxygen. I think this is the good option. If you have the big incubator, do all this, another step this, but the culture you can do in this small incubator, because you cannot change all the time inside the oxygen, the stress oxidative, temperature, and nothing this.
Yeah, Dr. Yeda, right. Is Dr. Sanjeev told about this point that at Transova, they don't use this
Yes.
bench top, and definitely now actually we request you with your research team, you come out with a day on which you can give with your media, with Reproduction media, to us same pregnancy rate with a DT and fresh embryos. That will be the great day. That will be very good day.
Yeah.
Okay, next question is again from Dr. Mutharao. Is there any donor effect? He is asking, is there any donor effect in the pregnancy?
Yes.
Okay,
Yes, you
Dr.
have.
Mutharao, it was discussed, yeah.
Yes, you have it. You have the donor effect in the pregnancy, some donor, and I can add more information. Not only the donor, but the donor and the bull, the interaction. Because some donor, I use it in the past during 6 months. One donor. And this donor produce a lot, almost 4 and 5 embryos with the bull one. And I work half of the year to do this with Brahma. And transfer 5, 4 embryos, nothing the pregnancy. Then when I change the bull, I transfer 5 embryos and give 3 pregnancies. Then you need to study this. You need to study because if you don't have pregnancy with this donor, maybe you need to change the bull.
Yeah,
And
there is one interesting good question from Dr. Siddharth. He is asking, in IVF program, when you take a new bull, should we first take trial on slaughterhouse oocytes before you take it for the regular IVF program? Do you advise that?
Here in Brazil, it is because you have the slaughterhouse ovary. Or then when I start a program, a big program to produce the embryo, then I test 5 or 10 bull in this farm. I test this with slaughterhouse ovary, and then I choose 5 to work during the project. But if you introduce a new bull, for example, if you have some donor that you know they produce the embryos, the pregnancy, and produce a lot of oocyte, then you can continue the half of the oocyte with the same bull and another half of the oocyte, the new bull. Then you can compare the production with this new bull. I don't know.
Yeah.
Sometimes I do this because if you don't have slaughterhouse oocyte.
We can do this in buffaloes in India, but we cannot do it in cows. Yeah.
Yeah.
Buffalo, we can do it, this trial. Now,
Yeah.
one question, when you select the donors, do you take even heifers as a donors, which are, say, having genomically tested? Do you select heifers as a donor?
You start, you have the program in the IDZE here with Nelore to produce the embryo, the heifer with 10 months, 20, and cows, and compare the production. And of course, if you have it with the genomic, they select the heifers, because here in Brazil, in United States, you use a lot with this in the milk. Select the heifers with genomics and use this to produce these embryos. Yeah, but the heifers produce less oocyte, and the pregnancy is lower than another.
Yeda, there is some people, they say you should, this is a question from me, you observe the cleavage on 4th day, and some people, some scientists say not to disturb them. Whether to observe, we should observe cleavage on 4th day or not to observe. What is your opinion?
Sorry, I don't understand. What is this?
Cleavage, should we check on 4th day, yes or no? Should we check the cleavage by, should we do it or not to do it?
Yes, I do this. I do when I culture of the embryos in the small incubator, and I use more medium, like in 4-well dish with 500 the medium, the IVC, I check very quickly. Because sometimes when you put the embryos to culture, you have some cells around of the oocyte, and these cells can attach in the plate. Then I saw very quickly the cleavage and the pipette to move this. Then but I don't stay long time to do this. To count, I count more or less. 8%. It's good cleavage or not good? Yeah, because it's only to see if the fertilization is good or not. Because sometimes you use some bull that cleavage it's a half. It's less than 50%. Then this is the problem, the IVF, the semen. But I saw very quickly in day 3 at afternoon or day 4 morning.
Yeda, in HF, Holstein Friesian, in HF cows, particularly, we see, you see a good cleavage. This question by Amol, we see a good cleavage, but after cleavage, these potential zygote, PZ, they get arrested. There is an arrest. They don't develop in Holstein. What are the reasons for that?
Of course, you have some reason that the oocyte don't have competence to development after cleavage, because when you have the cleavage until 8 cells, all the nutrient of this oocyte is this oocyte. After 8 cells is a activation of the DNA, the embryonic. Then if the media is not okay and the maturation of the oocyte is not okay, then they stop in this stage, the cleavage, and don't go until model or blastocyst. This is when the competence of the oocyte, but you have some problem. You have some problem. They stop the development when you use some premixture of the gas is not good. I saw this because if the premixture of the gas is not good, it's toxic. It's like it's a toxicity of the gas, the oil, and the serum fetal bovino in the medium. This is the 3 topics that can dead the embryos, and this is very slow, very slowly. It's like a homeopathic doses.
Okay, there is one question from Dr. Kailash Kadam. He is asking what the day of OPU or day of IVF, what should be the day you should consider for transferring embryo in recipient? Is it day of OPU or day of IVF? For transferring embryo in recipients.
Oh, the synchronization.
Yes.
Yes, the synchronization. Yes. The synchronization, I put the synchronization of the recipient, the estro. It's necessary to compare with the day of the IVF, because the IVF, I put the day 0. This is the day 0, the day the estro of the recipient, and day +1, the OPU. And if you compare, if a recipient have the estros in day the IVF and transfer embryo day 7, then the protocol is day 7, is the synchronization 0. It's okay, the synchronization. The synchronization +1 if the donor, if the recipient have the estro in day +1 in the ovum pickup. Okay, and this is good. This is good too. This is the protocol day 8. Okay, then the synchronization is 0, +1, and +1.
Okay.
I prefer the 0 and +1, day 8.
Yeah. Instead of negative synchrony, positive synchrony is better. Positive synchrony is better than negative synchrony. Right, Yeda?
Why? This
positive synchrony plus is better than minus. Am I right?
Yes. Minus no, +1 is the synchronous, it's day 1. The embryo is day 7, the embryo, and the recipient day 7 too. This is the synchronized 0 and the +1. But I think it's a good, when you start the IVF in 24 years ago, you synchronize like the flushing embryos, because the flushing embryo, you take the embryos in day 6 and day 7 flushing and put in the recipient +1, sometime +2, and 0, 0 +1, and +2, and not +1. Okay, and in IVF, we saw this is the contrary, this is the opposite, because the embryos develop more quickly than moat, than flush.
Thank you.
Yeah,
Which
Pietro tomorrow you talk a lot, and Pietro understand a lot of all the synchronized, the donor, recipient, and the protocol and everything.
yeah, tomorrow's talk will be very interesting with Professor Pietro Berselli. Yeda, which media you should use for carrying day 6 and day 7 embryo? Which media you recommend? Which nutrition media?
Some people like in Newman and some lab work to change the medium, the day 5 in 2 steps. The medium until cleavage, it's cleavage medium, blast medium, okay? And it changes something or put some another drugs in the medium to diminish, to not increase the lipids, yeah. But I don't use this. I use only one medium that I use all this substance in the since day 1. I don't change the medium, but sometimes I know some people that change the medium, they say to day 7. But now I think the person that work in the single media, all the process of the culture.
There is a difference. Some scientists say in your lab for cleaning your surface, for the laminar flow and all that, you don't use 70%.
Hi,
Should we
Dr.
Shah? Because some scientists, they say not to use 70% alcohol in the laboratory at all. Alcohol, you got alcohol. Yes. So 70% alcohol in the laboratory to clean the surfaces. Should we use or not to use? What do you say?
I use. I use alcohol. I use alcohol 70% to clean all the superfice, all the flower laminar, okay, and to maintain all the lab. But of course, if I have, I do this before to start the work and after the work. Then all the time in my lab it's clean, but not during because some people you go to the incubator to open the door and put the alcohol in the hands. No, in the hands. No, I cannot use this all the time. I use it before the start of the work and after work. And you need to think one observation. When you have the benchtop incubator, the chance to have some contamination with alcohol, perfume, or everything is can see volatile in the oil, and it's a problem. But the incubator, the small incubator, use only the gas inside the bottle of the gas. Don't use the gas, the air. And when you have a lab in Australia, you have in the time a lot of it's very hot, and sometimes the big incubator that use the gas, the ambient of the gas, you have more contamination in this incubator than the small incubator, because the small incubator don't use the gas in the ambient. It's only the gas inside the bottle, the cylinder of the gas.
But it is said that normally in our labs in India, we are using USAFE.
Yes, true.
That's
Yeah,
it.
but when you use the U-safe, you use it, for example, the CO2 that you enter in the incubator, but the another gas inside the big incubator is the air. It's not the bottle, only the CO2. But yeah, do you understand?
Yes.
Yeah.
There is one question from Dr. Vinod Patil. You know him, Vinod Patil. He was in JK Trust.
Yes.
And he came with me to Brazil. Recently, they have received embryo shipping media. What is ideal time to ship embryos in the straw in micro-Q incubator?
Uh-huh.
I am not, Dr. Patil, can you please ask this question? Rajan, इनका जरा वीडियो ऑन करो, Dr. Vinod Patil.
How many hours?
Okay, sir.
This is how many hours the embryos can be carried in the straws?
Yes. That's the question. Yeah. That is the question. Correct. Yeah, Yeda, that is the question.
Yes, this is the question. How many hours the embryos can be? If you use the medium, hold media, the embryo transfer media with hips, then you can use the 8 until 10 hours with this medium. Okay, if you have it, because if you can use it to put the embryos, of course, if you put it in the day 6, 1 day before, if you can use the vial and put the gas inside, it's better, but you need a technique to do the fill the straws in the farm. But if you don't have this, I think until 12 hours, it's enough. And the temperature that you use, I think the temperature, the transportation, this is 36 or 37.
Yeda, there are so many questions and it's almost, we are touching to 2 hours now. So what I will do, rest of the question I will email to you. I will send by email and I request you to reply, and I will inform all the participants we have asked this question to save time, because so I will email these questions. I'm sorry we won't be able to get a reply to all the questions because we're almost touching to 2 hours. So participants, kindly bear with me. Yeda, I have one question, very important question, is that your media is being used in the country almost in every laboratory, Reproduction. Now, we request you, because your present expiry of the media is 2 months, 60 days.
2 or 3 months, yes.
Yeah, can you try with your research to increase that expiry period? First question. And second question, in India, we are only in a young stage of IVF. We are not like Brazil. You started in 1998 commercial ET in Brazil, which is almost 24 years back. In India, we are very young, very young. So we request you to bring down your prices of Reproduction media, to lower the price. That is my second request. So expiry, increasing the expiry and reducing the price. These are the two requests from me as a national mentor for the IVF labs of the country.
No, you can, Dr. Shan. No, of course, you can do this. You can do this. You can do this. I can, because I believe now in United States, I have a place that you have all the mediums that I send each month to United States in the WTA. The office of my brother, and
Okay.
I send each month I send the media there. And sometimes when some media it's like maybe 2 weeks to expire, then you send it to university, you send it to another person to test the media, because I know here in Brazil you use this media expired, it's produced the embryos, yeah? Then, if you have some lab, if you can receive this medium, but I need to return, of course, the information about the produce this, because I do like that, I need the pregnancy, of course, the pregnancy rate of these embryos that you use with this. Then you can talk about this and reduce a lot the price and the medium that
Thank you.
expired.
And
Okay.
Yeda, I again request you, I mean, you have already told very important points, but can you please take 5 more minutes, and tell us as to how do we get good pregnancy rate from DT embryos? Please tell us some very, very important points you already covered, but again, please tell how to get good pregnancy rate from DT embryos. Some five or six important points, very, very important points. Please tell us for everybody.
I think to have this result, the pregnancy, DT, if you have this table, 40% of the pregnancy in DT, it's enough, it's good. But I think that you need to have a system of the culture of these embryos good, with lower oxygen, because I tested with this and if you have some lab that produce and have a good result, the pregnancy DT, use the lower concentration of the oxygen. Only not in benchtop incubator, but in big incubator too. Okay, but in big incubator, it's not so good to stabilize very quickly all these parameters. Okay, and I have a result in United States with this, with the good result, 50% of the pregnancy DT. But I need to do, I think the next travel to India, I have more results.
Okay.
And the results is the DT.
If you start giving us good results of DT embryos, it will be very, very good. And so, Yeda.
Sir, I have a small query, not query, a suggestion actually, sir.
Yes, Siddharth, I think you asked me that about tying up with something, no, the companies, right?
Yes. So, is there any possibility that
That can be asked
you
later.
collaborate with any Indian companies who can produce the media in India? because what is happening that when you import this media in India, 33% import duty is getting added. So if you can produce the media in India with collaboration with any Indian company, we can search for the companies that which can collaborate with you. Is there any possibility of such collaboration?
Yeda is asking, you got the question because you can let us know later also, but if you want to reply now also, you can think of, he's suggesting, Dr. Siddharth, you can tie up with some Indian companies. You know, there are a lot of media making companies in India. So that whether you can have a tie up, whether you can have a tie up with someone who can make a media for you and you can sell it, so we can save that 30% duty. That is the question. Siddharth, am I right?
Yes, sir. Yes, sir. Exactly. And
yes, Siddharth, go ahead. Please go ahead.
yes, sir. Yes, sir. That's my question.
Yeah. Yeda, you would like to answer this question?
To produce the media in India.
You can come and stay in India.
No, I don't know now. I cannot do. No, it's not possible. Of course, it is possible to do this. Of course, it is possible. But oh, sorry.
Bless you.
But now, thank you. But now I have all the parameters here to produce this, and I like to produce a lot of medium and control everything, of course. But you can do something together. If you have some problem, the logistics and this, I think that if you have some partner to receive like the media, and I send all the media, I know, because to send the media to India is very expensive, the logistics. Then you need to use the same logistics. I use the same, because the price to send one or two kit of the media is the same to send 10. Then I need to send all together in this place and distribute. I think that you can do this year, and maybe you can do something together to produce this media and in India and distribute, of course, the another place.
Thank you.
It's open the offer. I'm not closing of this. And thank you, Siddharth.
Thank you very much, Yeda. And as the end, we come to the end of the question and answer session, which comes to an end. So on behalf of the members of Bovine Embryo Transfer Group of India, I express my sincere thanks to you for making an excellent presentation and covering the various aspects of in vitro embryo production. I also thank all the members of the Bovine Embryo Transfer Group and also other participants from all other parts of the world who have joined this webinar. And thank you very much once again, Yeda, and have a nice day in front of you. Thank you very much. So with this note, we are not very keen to close this session because we have got so many questions, but you have given almost 2 hours to us, and we will send you these questions, and we request you to send an email. But for the information of everybody, you can note the Yeda's mail. I will send it to you, and you can always ask questions to her, and she's always ready to answer the questions. So thank you very much. And with this note, I declare this webinar closed. Thank you. Thank you, Yeda. Namaste.
Thank you.
Namaste. Indian way of greeting. Namaste. Thank you.
Namaste. Thank you very much. I'm glad to participate in this webinar and send the question to my mail, and I can send some article or some information for everything.
And Yeda, I have one more request. Society Brazilian Embryo Transfer